Hello friends, today we will learn about antigens. So let's begin. Substances which can induce an immune response, humoral, cellular or mixed T and B cells when introduced into an animal are called immunogens or antigens. Proteins, that means polysaccharides and nucleic acids, are effective antigens. Now let's know about the antigenicity of molecules. The immunogenicity and means we can say it as antigenicity of a molecule is not an inherent property but is dependent upon the experimental conditions of the system. Thus, the antigen, the mode of immunization, the species being immunized, and sensitivity of the method of detection are all important. Although characteristics of antigens are complex, certain conditions must be satisfied in order that a molecule be immunogenic. These are we will study. These are first point is only molecules that are foreign to the host are immunogenic. Second point, a molecule must possess a certain degree of complexity to be antigenic. Immunogenicity increases with structural complexity. In addition, aromatic amino acids contribute more to immunogenicity than do non-aromatic amino acid residues. Random polypeptides containing tyrosine are better antigens than the same polymers without tyrosine, and the immunogenicity of these polypeptides is proportionate to the tyrosine content of the molecule. As a general rule, molecules smaller than molecular weight 10,000 are only weakly immunogenic. Macromolecular proteins with molecular weights greater than 1 lakh are the most potent immunogens. The ability to respond to a particular antigen varies with the genetic constitution of the animal. Example, inbreed strain number 2 guinea pigs respond to poly L lysine, but strain number 13 guinea pigs do not. The ability to respond to antigens is inherent as an autosomal dominant trait. Now let's study about the structure of antigens. The initiation of immunoglobulin production requires binding of antigen to the lymphocytes surface. Only restriction proportions of antigenic molecules are involved in actual binding with antibody combining sites. These areas are termed antigenic determinants or epitopes, and they determine the specificity of antigen antibody reactions. Antigenic determinants are usually regions that project from the antigen surface. The antigenic determinants or epitopes are the smallest unit of an antigen to which an antibody or cell can bind. For a protein, an antibody binds to a unit which is about 3 to 6 amino acids, whilst for a carbohydrate it is about 5 to 6 sugar residues. Therefore, most large molecules possess many antigenic determinants per molecule. They are multi-determinant, and however these determinants may be identical or different from each other on the same molecule. For example, a carbohydrate with repeating sugar units will have several identical properties. Or determinants, you can say. And whilst a large single chain protein will have many units, each with a different amino acid sequences and thus many different antigenic determinants. Although the linear sequence of the residues in a molecule have been equated with an antigenic determinant, the physical structure to which antibodies bind are primarily the result of the conformation of the molecule. As a result of folding, residues at different parts of the molecule may be close together and may be recognized by a B cell or an antibody as part of the same determinant. Thus, antibodies made against the native natural conformation of a molecule will not, in most instances, react with the denatured molecule even though the primary sequence has not changed. Now let's know about the haptens. What do you mean by haptens? Very small molecules which can be viewed as single antigenic determinants do not stimulate antibody formation. These haptens, as they are called, can be attached covalently to larger molecules, means carrier molecules, and in this physical form are able to induce the formation of antibodies with the help of T cells. Carl Landsteiner prepared haptens by covalently coupling diazonium derivatives of aromatic amines to lysine, tyrosine, and histidine residues of immunogenic proteins. These proteins were called carriers. The protein hapten conjugates induced the formation of an antihapten antibody which was specific for the Azo groups on the protein hapten conjugate. The conjugate haptens thus behave as a complete antigenic determinant of the molecule. Finally finished. Any questions regarding antigen you can ask me in the comment box and subscribe my channel to know more updates about biotech topics. Thank you.